Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Ahmad Farihin bin Mazlan from 80-220-4D. So today I want to represent about my topic is transgenic approaches to microbial disease resistant in crop. So thanks for my lecturer Dr. Fatima, and let's begin. Okay, for the introduction. Disease causes by bacteria and fungi are currently some of the major factors limiting crop production of worldwide. In addition, it gives a negative impact on yield. Disease can also impact the post-harvest quality of food for reason, of course, efficacy and environmental concern. Much research presently aimed at transgenic expression of gene that can confer significant level of disease resistance. Although the recent introduction of plant product for control of insect pests have been highly successful, transgenic plant exhibiting resistance to fungi or bacterial diseases have yet to reach the marketplace. In this review, we summarize research result with implication for developing disease resistant transgenic crop. Due to a space limitation, we will focus on control of fungi and bacteria. Much work in this area center are understanding natural occurring plant signaling pathway that control pathogen resistant and identifying and cloning gene encoding antifungal protein. We highlight recent development in both of these areas. Okay, so for the gene, they use a R gene or resistant gene. So the cloning of a large number of these genes has shown that R gene product shape motif such as lecherous lipid or LRR, nucleotide binding site and kinase domain consist with their roles in signaling pathway and suggestion common mechanism for pathogen recognition which might be exploited for applied goals. So the cloning of the flax M gene controlling rust resistance showed that mutation of leading to loss of R genes function mapped to the three region encoding the LRR. Alright, so for the method, most of reported dominant disease resistant genes in plant R genes encode NBLRR immune receptors. Plant genomes carry many NBLRR type R genes that recognize specific pathogens and induce resistance against them. Thus, this immune system in plant is tough in perform similar function as the adaptive immune system in animal. Okay, so for the result, we get a transgenic expression of antifungal proteins. The enzymatically active antimicrobial protein include chitinase, glucanase, and lysozymes. Chitinase and glucanase are capable of regarding fungal cell wall components, and in vitro, some of these enzymes display strong antifungal activities. So genes for this or other enzyme have been introduced into a transgenic plant with varying rates of success. Okay, so next is we get a gene of attack and defense. So toxin may act as a virulence factor in activation of this toxin or their target in the plant has a lead to enhance disease resistance. Disease control in this system may include engineering incentive variants of toxin targets in the plant, expression of toxin inactivating enzyme or blocking entry of this toxin into the plant cell. Right, so for the conclusion, rapid advancement in our understanding of the genetic mechanism behind this resistance in plant have created a variety of novel and interesting prospects for pathogen control engineering. These possibilities can now be investigated in the near future thanks to the tools made possible by the cloning of R genes and other signaling pathway elements like NPR1, NIM1, or MIO. We can build a new broad-spectrum defense mechanism in crops by combining the understanding how this protein works with the rapidly developing knowledge of other natural defense process. Combining this defense with the additional protection offered by the expression of powerful antifungal proteins ensure the crop grown will eventually have access to a potent arsenal to fight off the most significant microbial illnesses currently restricting crop productivity.